All right, thank you to the organizers for the chance to present some of our work on SARS-CoV-2 variants and their potential biological implications. I want to disclose that we have patented the infectious clone system that we use in these studies and a number of other studies, and so we have those financial interests going forward. And so briefly, what I'm going to talk about today are the SARS-CoV-2 variants and give kind of an outline of where we are with these variants. Well, obviously, it's been a topic of great interest over the last couple of weeks, and that importance isn't going to change over time. And so I'll talk a little bit why it's important and then what drives these variants. And then we'll outline a couple of the critical COVID mutations that have been observed in some of the new variants over the last couple of months, and then how we're going to test the impact of these variants. And so why are these variants important? So to really define these variants, really these are small changes that may increase or augment the virus's ability to increase its ability to transmit or replicate, or alternatively escape from immunity. Now, this isn't common in coronaviruses, or is less common than other RNA viruses because coronaviruses encode a proofreading mechanism. So the changes that we see are not primarily driven by error-prone polymerases, but instead they're accumulated mutations that happen over time and often are associated with fitness advantages, although not always. And this is really going to be a question for SARS-CoV-2 going forward. It's in the population, and so there are going to be pressures on this virus to become more fit at replication and transmission states, as immunity comes online through vaccines and natural infection. You're going to see more and more variants that can escape or partially escape from these protection, become a little bit more fit. And so this is something that we have to really consider moving forward. And so there have been a number of variants of interest over the last year or so of this COVID outbreak. The first was the D614G, which I'll talk a lot about today in terms of examples. But most recently, three variants, the UK variant, the South African variant, and the Mink variant have made a lot of popular press and have been causing a bit of worry around the world. Most recently, in the last couple of days, there's been a newer strain from Brazil that has similar elements and may be the next important variant that comes and starts to emerge in the human population. And so the importance of variants really focuses on the spike protein. It's the most common and important target that has most of the changes that we're observing. And the reason that this is important is that the spike protein for coronaviruses is a critical key for infection. It's needed for both virus binding to the cell, host cell, and for entry. The other aspect is that this is also the main target for immunity, and the changes in the spike protein can alter the ability of an antibody to bind. And so changes in these variants are really important and something that we really observe and take great stock in. Now, there are other viral proteins in these variants that do change. And these are more associated or often more associated with host interaction or viral processes. And these are really potentially very important, but harder to interrogate. And so while groups will start looking at this, these are not necessarily the first mutations that people will explore when studying these new variants. And so shifting gears, what drives these variants? And so really we can divide this into kind of four parts. The first is replication and transmission advantage. This is kind of the simplest idea. Viruses that replicate better are going to have a fitness advantage and become more dominant over time. The idea here is faster replication, but faster entry, they may be more easily transmitted. And so as they replicate in the population, they become the dominant strain. Examples of this are a furin knockout virus that my lab generated over the last year, and then this D614G variant that's been described in the literature and work that our group has done. And so the idea is that when we actually had generated a mutant virus where we deleted the furin cleavage site, which is a key component of SARS-CoV-2, what we found is that this virus actually replicates better than the original wild type in a certain cell type. These are called vero cells. This is an amplification vero cell type. And what we find is that when we put the wild type and the mutant and we compete them, adding them at a ratio of 1 to 1, within 24 hours, we can see that in the blue bar, we have much more of the mutant than the wild type. Now, this shows that there's a replication fitness advantage for this mutant, but only in these vero cells. When we took this into in vivo animals, we found the inverse was true, and that the mutant, this loss of this furin site, resulted in an attenuation of the virus. With a D614G mutant, we did the same thing where we made a infectious clone that had a single point mutation. We took the D614 amino acid and made it a G. And what we found is that this virus actually has this G virus, this G mutation has a fitness advantage. When you look at in hamsters, and these are in different tissues in the hamsters, one being if they're completely equal, you can see that there's a small trend that there's an advantage for this D614G mutant in animals in all of the tissues we looked at, most notably in the upper airway tissues. When we did the same study in primary human airway lungs from humans, what we found is that, again, we see a small advantage, and over time, this advantage increases, really illustrating that this one mutation has an advantage that allows the virus to replicate a little bit better, and in other work I'll talk about later, transmit a little bit better, which is why this D614G mutation has become the dominant mutation or the dominant isoform or strain in the population now. A second aspect that drives COVID variants is this immune host modulation. And this is a change that can alter interaction with host factors, whether it be the immune system and factors like type 1 interferon or interactions with key host components. This includes components that may be involved in translation or taking over the virus or the host cell. And we don't normally associate these with spike. The spike protein, while it has a key role, is not really known for host modulation. So I won't talk much about that today. The last is immune evasion and escape. And these are mutations that allow escape from antibodies and other primary immune system memory systems. The idea here is that if you have a deletion or substitution in the spike protein at a critical residue, this residue might be where an antibody binds and actually neutralizes the virus. Usually this selection occurs when a population has immunity. And so we're not really expecting to see that just yet, or selection based on immunity yet, because we're not near herd immunity. And the vast majority of people have never seen COVID-19. As vaccines come online, that'll start to change. The examples we have here so far are this E484K and S477N. These are mutations that have been shown to escape monoclonal antibody treatments and may be important for treatments and other escapes from immunity. And so the idea here, and this is an example from this Andreo paper, where here E84K, that mutation results in an almost 50% drop in the neutralization titers. The titers don't go down to zero, but the incorporation of this mutation here, passage 12, really allowed this virus to escape polyclonal sera, which is obviously an important protection when we consider vaccines and natural immunity. The last thing that we think about with variants is that there are non-specific founder and carrier mutations. What does this mean? Well, there are mutations that occur just naturally. While coronaviruses have a low error rate, these mutations can occur just by accident and be carried along. And so they may not have an impact on infection or replication. They just may be something that happened in the course of replicating in the variety of hosts that it goes through. The example of this is that there are two other mutations found with this D614G. This P4715 in ORF1A and this G204R in the N protein were carried along, and from what we can tell, that they don't have any significant impact on pathogenesis or transmission in the studies that we've done, really arguing that they are a founder mutation that don't have biological implications. And so shifting gears, what are the critical mutations that we've found so far in these COVID variants? And so just to orient you, this is the COVID spike protein. I'm divided into different domains, here 4 domains in 2 large parts. And so the top part contains the NTD, the N-terminal domain, and the RBD. The N-terminal domain has been associated with attachment to cells. The RBD is the critical component that binds to the human receptor ACE2. The second aspect is that this portion forms this globular head that gives coronaviruses its spike. It's also the most variable portion of the spike protein and has a certain amount of plasticity to it. In contrast, the stock portion contains the CTS1 domain in pink and the gray machinery of the S2, which is the fusion machinery. And these are highly conserved across the coronavirus family, and they're required for entry and activation of the virus. And so both parts are important, and each of them has different areas that can change and be altered over time. as these variants approach. And so when we look at the variants and the mutations, we kind of can divide into these six kind of classifications. In the first block, we have the UK variant, and these are the mutations that are found in the S1 or the CTS1. This is the highly conserved domain. We also have the South African and the Brazilian variants. We've also layered here the mink and mouse adapted strains, and then I've identified single amino acid changes or deletions that are associated with escape. And so when we look at this, the vast majority of virus changes in the CTS1 S2 region are really in a highly conserved region. There are only two that are on the exterior portion that may have any impact on antibody binding. But for the most part, these are all related to the machinery of infection and entry. And so it's not obvious that these will have major implications for infection. That being said, the D614G mutation has already been shown to be in 3 variants, and that's really indicative that it's important. And importantly, we've shown that it plays a role in transmission, in the mechanism of the isoform that forms and how the orientation of the spike, the new orientation with the G mutation allows the spike to be more easily cleaved and more easily bind to the receptor, allowing it to transmit a little bit faster. Looking at this a bit closer, we focus in on the N-terminal domain. This is the region that is the most highly divergent region of the spike, and it's been associated with attachment with certain coronaviruses. And so what we find is that for the most part, these mutations are all on the exterior surface of the N protein, many in prominent positions. You can find it on the backside here too. What's interesting is these mutations are mostly external, and so they may have an important impact on antibody binding, and they may play a role in attachment. Interestingly, this delta 6870 deletion, this particular loop here in the cyan color, has been identified in 2 different strains, in the UK strain and in the mink variant, really arguing that, again, we've seen this in multiple locations. You don't see multiple deletions in any of these other variants, arguing that there may be selective pressure at this delta 68 that's driving this mutation and have biological implications. The mutations that we are most interested in are in the RBD, though, the receptor binding domain. It's here is where we find a number of mutations. So the RBD is this red portion on top. When we focus in on the receptor binding domain, this is the binding cleft between humanase 2 and the dark gray. And here on the light gray, we have the spike protein. And these are each of the key mutations and their location in that RBD binding cleft. And what's notable about these is that obviously changes here in antibody binding in these locations will have major implications. And while there's plasticity here, you can only have so many changes here without disrupting antibody bind or receptor binding. And so it's kind of a bit of a cat and mouse in that you can have changes here, but you may lose fitness by losing antibody or receptor binding. At the same time, if the antibody binds there, those viruses can't replicate. And so these mutations here in RBD are our primary interest and what we'll really focus on, most people are focusing on in terms of examining these variants. And so how do we evaluate mutations in the spike protein in these variants? And so what we find is that, and we predict that most of these spike mutations won't modulate host immunity. They're not going to change some kind of interaction with the host outside of the receptor. We do expect that we'll see alterations in fitness or transmission, and that can both be positive and negative. A third aspect is that antibodies binding can change. And so the RBD and different parts of the spike, this is where our primary immunity is. And so any changes here can have important implications for immunity kind of long term. And so in looking at these, we've really identified 3 mutants that have been common in these variants that are of concern. This 501 mutation here at this location in blue is found in 3 variants. It's found in all 3 human variants, and it really argues that it's playing a role based on its location in interaction. It's in the 3 human variants in UK, South Africa, and Brazil. It's also found in a mouse adapted virus. So it really argues that it has an ability to alter binding to humanase 2 and change the dynamics of transmission and replication. The second mutation is this 417, which has been described as an N in the South African strain. It's also been described with other residues in Brazil and the mouse adapted strains. So it really argues that this variant also, this mutation can play a role in interaction with the host and may have implications for the ability of antibodies to bind. The last one that we think is probably very important is this E484K, which is again found in two variants, South Africa and Brazil, but has also been associated with escape mutations in polyclonal sera and monoclonal antibodies. And so this really argues, based on its location and its interaction, that it's playing a role in antibody binding as well as potentially in binding receptor and transmission and replication. And so the question is, what will we be doing to test these? Obviously, we're still early on in these studies. Really, over the last month or so, these have become much more prominent, and we're still in the process of generating and working on these reagents. But we have examples for testing this, and we kind of look at this in a variety of ways: in vitro, and for transmission, which is part of the in vivo studies. And so the best example of this was that as this D614G variant became more prominent in the population, we wanted to know what was the difference between D at the 614 mutation versus the G at the same position. And what we did first is we examined them in Vero E6 cells. Again, this is an amplification cell line, and we found no real distinct difference in viral titer, genome load, or RNA PFU ratio when we compared them in this specific cell type. In contrast, when we looked at them in primary human airway cultures, again, as I mentioned earlier, we see that this G mutation allows an advantage, and that advantage over time increases, really arguing that there's a replication advantage that drives this mutation. And we can see that fitness in this cell culture dish model of primary human airway cells. When we're shifting gears, we've also done these studies in vivo. And so we've taken the hamster model, and we can see that in terms of overall disease in panel A, we don't see huge changes between the D and the G mutant. They're very similar in the total amount of disease. And the loads are relatively similar, although you'll see that in the airways, specifically in the nasal wash, we have higher loads for the G mutant relative to the wild type D strain, really arguing that, again, these small changes in specific tissues are probably contributing to the advantage this variant has in transmission. The other thing that we found is that when we directly competed, so when we added the same amount of the wild type and the G mutant to a hamster over the time course, what we find is that there's a small advantage for the G mutant. It outcompetes the wild type and really shows that it has an advantage in animals in all the tissues that we tested, really arguing that it does have a fitness advantage, and that's what's driving its emergence in the human population. We also found that the G14 mutation, this is work that Ralph Barrick's group did, where they co-housed hamsters and ferrets and asked whether the G or the wild type mutant transmitted better. And what you can find looking at this last panel on the right is that there's more transmission, higher loads in the exposed animals exposed to the D mutant than they are to wild type over time, really indicating that there's a small advantage for D614G in that population. And so none of these studies have yet been done for these more recent variants. We've actually started these studies with hamsters with 501, the N501 mutant, the single mutant. We've also made a double mutant in our infectious clone system that has the deletion of the 6970. And we're preparing to do these same studies with the individual variants. We have access, and we're getting the UK and South African variants. And we're making a number of additional mutants that are being prepared now. The last aspect is testing for antibody escape. And what we're interested in is how these variants work and examine how well acute and convalescent sera from a recovered patient works against them, as well as vaccine sera. And so previous work with a D614G found that in general, the G mutation doesn't really alter our antibody binding. There are small changes, but for the most part, we see strong neutralization whether the virus has a D or a G at position 614. In terms of the variants that have come out so far, the positions that are most noteworthy are N501Y, E484K, and this 417N, both of them a combination of their ability to transmit as well as potential for antibody escape and escape from the immunity that's been generated. Lastly, we are testing all of these ongoing. Us and other groups are all around the world, and I expect in the next few weeks we will have many of the answers we're looking for some of these, for many of these variants, and the impact they'll make long term. And with that, I'm happy to acknowledge the group, my group. Brian, Kumari, and Craig have been doing the majority of the work with the variants here. Pei-Young Shi's group has led most of the studies with the antibodies, as well as making variants in their group. We've gotten a lot of support from the World Reference Center uh, at UTMB for these studies, and then funding from NIH through an R01 and a U19 system. Thanks.